This is the last OCI level biology video on Unit 2, and this one is Unit 2.4 Enzymes, and this video is going to cover all of this, looking at different effects on temperature, pH, changing substrate levels, etc. on enzymes, and we'll finally end it with looking at some toxic drugs and their effect on enzymes. So first of all, a little summary of what enzymes are. So enzymes are biological catalysts that speed up metabolic reactions. They are unchanged by the reaction, so they cannot be used up. There's small numbers of catalysts that can catalyse large numbers of substrate. So a turnover number is the number of reactions an enzyme molecule can catalyse per second. And that's sort of how we measure how efficient an enzyme is. Enzymes can speed up reactions 10 to the power of 12 times within their optimum temperature, pH and pressure. And they don't create unwanted byproducts. They rarely make mistakes, and production can be regulated to meet the organism's needs. So let's have a look at their structure and function. Obviously enzymes are proteins, so they're built up of amino acids. They've got a primary structure, a secondary structure. They've got a tertiary structure as well. So, and this tertiary structure within it is going to contain those different bonds, those hydrogen bonds, all sorts of like ionic bonds, covalent bonds, disulfide bridges. Alright. So, if we look at this here, this is our enzyme. It's a protein tertiary structure, so it can have different shapes. So enzymes have got the body bit and the active site, and this bit that's not the active site we call the allosteric region, or the allosteric site, and this is our active site, which is where the substrate goes in. So the active site is usually complementary to the substrate. So large molecules with a specific area, about 6 to 10 amino acids long, big, forms the active site, and the tertiary structure gives the active site a complementary shape to the shape of the substrate. Each enzyme is complementary to its substrate and only its substrate. So active site, substrate, as you can see this enzyme, it's got a different active site, it's complementary to this substrate, and it will only be able to synthesize this substrate. As I've said, enzymes can break down and build up. So when they're breaking down, one substrate comes in, it's broken down, and it'll end up being two like that. When we're talking about building up, we usually have two substrates going. They temporarily bind together as well and join together to form one large product molecule. When the substrate is within our active site, we call it the enzyme substrate complex, and when the product is within the enzyme, we call it an enzyme product complex. When the product's released, we just call that our product. Okay, so I'll point that out later. And our tertiary structure of the active site gives it the complementary shape. So there's two forms of enzymes. We can have intracellular or extracellular. So intracellular speaks for itself. Within cells, for metabolic reactions such as, and these are known as metabolites. So metabolites are broken down into smaller molecules to release energy, that's catabolic reactions, or there can be an anabolic reaction to synthesize larger molecules from smaller ones, such as synthesizing ADP to ATP. Okay, and we'll look at those endothermic and exothermic energy curves to do with catabolic and anabolic later in the video. So this is a catabolic reaction, breaking these substrates down. This is an anabolic reaction, building things up. So I'll just speak to you about catalase for a bit. Catalase is found in nearly all living organisms exposed to oxygen. It protects cells from damage by reactive oxygen by quickly breaking down hydrogen peroxide. Catalase consists of 4 polypeptide chains and contains a heme group with iron. It is the fastest acting enzyme, with its turnover number being 6 million per second. It's found inside small vesicles called peroxisomes, and white blood cells use catalase to help kill the invading microbe. So it has a role to protect organs from damage by reactive oxygen, and that 6 million reactions per second is absolutely huge. That's what we're sort of dealing with here. We're not dealing with it goes in, it sort of cooks in this oven for a few minutes and it comes out broken down. No, this happens instantly, really, and several hundred thousands of times per second. Then we've got extracellular enzymes, and they have a function outside of the cell. And the main example of this is fungi releasing hydrolytic enzymes from their hyphae, which digest material from outside the cell. So they digest food, and then they absorb its useful products. But another example that you might think animals don't have extracellular enzymes, but that's wrong. Think of like amylase, producing the salivary glands. It acts in the mouth, digests starch. That's not within a cell, is it? Your mouth is not a cell. So it digests starch and maltose, and it does the same job in the lumen of the small intestine. And then we've got trypsin in the small intestine. Okay, and they have different optimal temperatures. So these enzymes all have different optimum pHs, optimum temperatures, optimum pressures, and we'll look at pH and temperature and its effect on enzymes later. What we've got here is cofactors. So cofactors are non-protein molecules that help assist the enzyme to help it work efficiently. So cofactors can be organic or inorganic. So an example of organic cofactors are coenzymes, which are chemically changed during the reaction by temperatures. And organic basically means anything that contains carbon in it is usually organic. Chemists might have a better definition for it, but you don't need to know how to define it. You just need to know that coenzymes are organic. An example of this are vitamins. So there's several vitamins that assist enzymes. So vitamin B12 helps cobalamin enzymes. You don't need to know what it does or how it works. If you look at nicotinamide B3, sorry I can't pronounce that, it assists NAD and NADP, and we see that in respiration and phosphorylation. So we also use B6 to assist coenzyme A, and we see that at the start of the Krebs cycle to form acetyl-CoA. Okay, so these vitamins are organic molecules that assist enzymes in their function. Then we've got these inorganic ones, and these are metal ions, such as chloride ions, which act as a cofactor for amylase. They do the exact same role as organic coenzymes would. Inorganic metal ions are a cofactor which help assist the enzyme. They usually would bind to an allosteric region, or they sometimes bind within the active site. But they help assist the function of the enzyme. Without them attached to them, most enzymes wouldn't work. So a prosthetic group is basically a cofactor which is permanently bound to the enzyme. Most of our cofactors are temporarily bound, and they are used up during the reaction. Some only last for one reaction, then they need another cofactor to bind to it. Others just bind for so long. But prosthetic groups are basically permanently bound cofactors, and an example of this is zinc in carbonic anhydrase, which converts CO2 and H2O to carbonic acid. So that reaction that's constantly coming up in all of our topics. If you don't know it, there might be several areas of biology you might suffer from. It's a very likely thing to come up, considered it's covered 3 times in the course. That's the example OCR want you to learn. All the examples I put on here I'd advise you learn. Maybe for coenzymes, in the specification it just said vitamins and their role. I've listed 3, I think there are 5 or 6 in the textbook. I'm just learning these 3. If you think you should probably learn 6, then go for it, have a look in the textbook. I ran out of space, so I put prosthetic group within this enzyme, which is basically what it's doing. It's permanently bound in the enzyme. So zinc in carbonic anhydrase. So definitely remember chlorine in amylase as a cofactor. You must remember zinc in carbonic anhydrase as a prosthetic group, but you should just remember vitamins help coenzymes work. Okay, so that was just a basic introduction of enzymes and how they function. Now let's look at the main hypothesis, so the lock and key hypothesis and the induced fit hypothesis. So in the lock and key hypothesis, they state that enzyme's active site is complementary to that of the substrate molecule. Temporary hydrogen bonds hold the enzyme and substrate together, forming an enzyme-substrate complex, and we'll just put ES complex from now on, or ESC. And the substrate molecule is broken into smaller product molecules that leave the active site. So 2 smaller substrates in an enzyme-substrate complex form bonds between each other, creating an enzyme product complex. The enzyme and the substrate molecules have kinetic energy and are moving randomly. If we have a successful collision between an enzyme's active site and the substrate, then we form this ES complex, the enzyme substrate complex. The substrate molecule is either broken down or built up, so we've learned that catabolic and anabolic. Once the product molecules leave the active site, the enzyme can form another ES complex. So while we've got this substrate within our active site, nothing else can react but that substrate, and until that substrate is reacted and used up and the enzyme substrate product complex deforms and the product is released, only then can we react. And for once in my life I've actually outspoken the text. So then we've got the induced fit hypothesis, and the induced fit hypothesis is slightly different from our lock and key hypothesis. Whereas lock and key, we're basically saying every enzyme has a specific substrate and it's an exact fit and it doesn't change shape, it just slits in like a key into a lock. Okay, that's exactly what is that. Must be the perfect fit. In our induced fit hypothesis, we're basically saying the active site changes shape slightly to mould itself around the substrate. So the book puts it saying like it's like a glove. So a glove on a hand. Gloves aren't perfectly shaped hands because all hands are different sizes, but the glove, these latex gloves, they mould around the differences in each hand. Non-covalent forces such as hydrogen bonds, ionic attractions, van der Waals forces, hydrophobic interactions bind the substrate to the active site. The product molecules have slightly different shapes, so they detach from the molecule, freeing the enzyme to catalyse another reaction. So we get the enzymes and substrate. This forms an enzyme substrate complex. Then it goes to an enzyme product complex, and then we get our enzyme and product, and it's free to react again. So our induced fit hypothesis is basically saying this active site actually can slightly alter its shape to fit around the substrate, and this is how I've drawn it here. This enzyme here, it's not really specific to these 2 substrate molecules. When they come in close contact to it, new forces cause the enzyme to mould around the substrate, and look how a tight fit it is there, allowing the reaction to take place. If we're seeing anabolic reactions of building up, the 2 substrate molecules form slight bonds between each other, usually hydrogen bonds, and then they bind together. Okay, so that is induced fit versus lock and key. Now let's have a look at our how enzymes lower the activation energy of a reaction, and what I'm talking about there. We've got this graph, and then I'll speak to you about this graph in a second. This is a reaction. This top line is a reaction without an enzyme. And I'll probably redraw on the line for you so you know which one I'm talking about. So without an enzyme, we see this curve. And this bit here is our activation energy. And that's the amount of energy we require to start the reaction, basically. The enzymes lower this activation energy. So if we use an enzyme and do this in green, if we see this curve, it only slightly, I didn't do that very well, it only slightly increases and then it follows the same trend. This is the important bit. The important difference is over here. So we've got, without an enzyme, we've got a huge amount of activation energy needed to start the reaction. With an enzyme, look how little activation energy is required to start the reaction. So enzymes lower this activation energy, meaning they increase our metabolic rate and rate of reaction. So this is an endothermic reaction, this is an exothermic reaction. This is not in the book, but it's something that maybe we get some suggest questions and you might have to suggest which one belongs to what sort of reaction. So an endothermic reaction is one that means the product has more energy than the original substrate because we've put lots of energy into it. And an endothermic reaction, something like that, is ATP. So ADP plus P forms ATP. I believe that's an endothermic reaction because we're building it up. It's an anabolic reaction. So most anabolic reactions are endothermic. We've got ADP and P, which have got fairly high energy, but they've not got much value for us in releasing energy. But when we bind them together, which takes a lot of energy, we can form ATP. And this ATP, as we know, when we hydrolyze it, has got huge amounts of energy, and that's why it's an endothermic reaction. So our product has more energy than our original substrates. If we use an exothermic reaction, so we could reverse that. So when we release the energy from ATP, this could be a catabolic reaction, and we see the ATP with a lot of energy. Then this is the activation energy, so our hydrolysis reaction, the energy needed to break down our ATP. Once we do that, we release all of its energy. Lots of energy is released to the external environment in exothermic reactions, and then we get this ADP and P again at the bottom. Okay, so that is how I interpret those graphs. Any chemist might comment if I'm wrong, or if ADP and P is not actually an endothermic reaction, but a pretty good estimate would tell me that it is.